students, in this video we are going to discuss about RNA as a genetic material. We already know that DNA acts as a genetic material and it carries information from generation to generation. And in this experiment, the scientists proved that RNA is also a genetic material. So first of all, we should know what is RNA. RNA is a ribonucleic acid that acts as a genetic material which carries genetic message from DNA to ribosomes and is involved in the process of protein synthesis, which is referred as RNA. It is one of the two types of nucleic acids found in all the cells. The genome of viruses may be DNA or RNA. Most of the plant viruses have RNA as their hereditary material, and this scientist Frankel Conrad in 1957 conducted experiments on tobacco mosaic virus, which is denoted as TMV, to demonstrate that in some viruses RNA acts as a genetic material. So the TMV is a small virus composed of a single molecule of a spring-like RNA encapsulated in a cylindrical protein coat. The image is shown here. There are different strains of TMV can be identified based on their chemical composition. The TMV infects the tobacco leaves, causing the infected regions on leaves to become discolored and bristled as shown here in the image. And there are different strains of TMV which produces clearly different inherited lesions on the infected leaves. For example, the common viruses produces a green mosaic disease, but another strain of TMV named horn's rib grass, which is denoted as TMV-HR, produces a ring spot lesions in the leaves as shown here. Moreover, the amino acid compositions of the proteins of these two strains differ. So that scientists Frankel Conrad and B. Singer first developed the techniques for separating the TMV particles into RNA and protein. They found that virus could be broken into component parts and they could again be reassembled or reconstituted to form functional virus. It was also possible to synthesize chimeric virus particles using RNA from one strain and protein from another strain. Such chimeras had serological properties of those strains from which protein was derived, but in other properties it resembled the other strain from which RNA was used. Let's move on to the experiment. The two scientists separated the protein and RNA from the two strains of TMV, that is TMV common and TMV-HR. Then they reconstitute the viruses with the RNA from TMV common with the TMV-HR protein and TMV-HR RNA with the TMV common protein. When these reassembled viruses were used to infect tobacco leaves, the progeny viruses produced were always found to be phenotypically and genotypically identical to the parent strain from which the RNA has been obtained. The reassembled viruses with the TMV common RNA and TMV-HR protein produced a green mosaic disease, that is the characteristic of TMV common strain. The recovered virus had a protein characteristic of TMV common. This proved that the specificity of virus protein was determined by RNA alone and that proteins carried no genetic information. Hence, RNA carries genetic information and not the proteins. And this image demonstrates that RNA as a genetic material. The scientists took two strains of TMV and named as TMV-A and TMV-B. In the TMV-A, they have separated the proteins and RNA and named as protein A and RNA A. And in the TMV-B also, they have separated the proteins and RNA and named as protein B and RNA B. Then they reconstituted the virus by mixing the protein A with RNA B and protein B with RNA A. Then they allowed to infect the two combinations, that is the reconstituted virus, to affect the tobacco leaves. In the first set, the leaf showed the symptoms that was related to the TMV-A, which contains the RNA of TMV-A, and in the second set the leaf shows the symptoms of TMV-B, which contains the RNA B. Hence, they concluded that RNA was transferred and not the proteins, so the RNA acts as a genetic material. Thank you.